Hello there. Welcome to Medical Microbiology. So today we will continue our lecture chapter 3. Today we will discuss about staining technique. Dyes are used to create contrast by imparting color. So staining is using dyes to create a contrast on the object that we see under the microscope. There are two main types of staining. The first one is the positive staining. So positive staining, this positive staining usually use basic dye. So basic dyes are cations. It means they carry positive charge. Just remember, the cell is actually more negative. So cell carry negative charge. If we stain the cell with basic dyes or positive dyes, and what happens? This positive will bind to the negative charge of the cell membrane or the cell wall. It means that the stain, okay, the dyes will stain the cell. So the cell will be stained if we use the positive staining, if we use the basic dyes. So this is examples over here. Okay, so this one, okay, so this one and this one are positive staining using basic dyes. So you see the cell is actually stained with the color. The cell stained with the color. The background is actually brighter because the background is not stained. So this is the positive staining using the basic dyes. The second type of staining is called the negative staining. It is using the acetic dyes. So acetic dye is an ion which is negative charge. And remember again, the cells, the cell membrane, the cell wall, they carry negative charge. So what does this mean? It means that this acetic dyes, which is negative, will not bind to the negative charge of the cell. It means that the cell will not be stained, but the background will be stained. Okay, so this is the negative staining using the acidic dyes. As you see over here, the one that gets stained is actually the background. So the background has stain. The background has stain. While the cell itself is clearer, is brighter because it is not stained by the negative staining, by the acetic dyes. Over here, the cells are white or brighter because what? Because they are not stained by the acetic dyes. They are not stained by the negative staining. So this is the two important informations about the two main type of staining, whether using the basic dyes or the acidic dyes. The basic dyes will stain the cell, okay, and the acidic dyes will stain the background. Okay, now there are some variations in staining. Okay. The first one is called the simple stain. So simple stain only using one dyes, whether basic dyes or negative dyes only. Okay, it's only using one type of dyes. This is simple stains. The second type is called different stain. So this one use at least two different dyes, different type of dyes. Usually the primary, okay, the first dyes will be basic dyes. Okay, so the primary stain will be basic dyes, while the counter stain, the second dyes will be acidic dyes. Example is when we perform the gram staining. So they use two types of dyes, primary stain and counter stain. The primary stain is usually basic dyes. The counter stain usually the acidic dyes. The last one, okay, type of stains is structural stains. This is a special stains to reveal the part of the cell. Okay, using special dyes to reveal the specific structure, whether inside the cell or outside the cell. For example, if inside the cell, okay, looking for some small structure inside the cell. If it is stain outside the cell, like for example, maybe staining for flagella, okay, so called the flagellar stain, or maybe stain for the capsule, capsular stain. So using specific dyes to reveal some structure of the cell. If we look at the result, maybe look like this. So this one is simple stain using only one type of dyes. In this case, using purple dyes. Okay, this is a crystal violet dyes using one only basic dyes. It will stain this cell in the same color. And then the second one, this one is different stain. This is actually gram staining. If you see the left side over here is positive. This one is positive gram, okay, gram positive. The color is purple or blue. And this one is gram negative bacteria. The color is red or pink. Okay, so this using two dyes, positive dyes and the counter stain. Okay. And then this is example of structural staining, which is using the special dyes to refill the capsule around the cell. If you see the line over here is actually the cell. It is protected by the capsules around it. So this type of staining reveal the capsule or showing the capsules of the bacteria. Okay, I believe this is enough for a staining procedure. We will meet again on the next lecture, which is media for culturing microbes. I'll see you and goodbye.